Okay, you said give me everything, so here we go. This is the no skips, all hits biology vocab mega drop.
And we're not reading definitions like robots, no textbook voice. We're gonna break this stuff down like we're explaining it to your cousin who thinks mitochondria is a Pokemon.
Let's start basic. Prokaryotic cells, they're like budget phones. No apps, no bells and whistles. Just the basics. No nucleus, no fancy organelles, just bacteria doing their thing.
Eukaryotic cells are the iPhones of biology. They've got everything, a nucleus that holds your data, plus membrane-bound organelles, which are like apps doing different jobs. Plants, animals, fungi, those are all eukaryotic.
Cell wall? Think of it like the armor on a medieval knight, or your house's brick wall. Found in plants and fungi, it gives the cell structure and keeps it from popping like a balloon.
Plasma membrane is the gatekeeper. It controls what goes in and out of the cell. Picture a nightclub bouncer with a checklist. Water? Yes. Giant toxic molecule? No thanks.
Vacuoled storage room, big in plant cells, stores water, nutrients, waste, like your closet that holds hoodies, snacks, and your emotional baggage.
Nucleus is the control center, your brain if you were a cell. It holds your DNA and tells the cell what to do. Inside that is the nucleolus, which builds ribosomes, tiny factories that make proteins.
Chloroplasts are found in plant cells. They do photosynthesis, which is how plants turn sunlight into sugar. Think solar panels that print candy.
Mitochondria, yes, the powerhouse of the cell, converts food into ATP, which is energy your cell can actually use. Basically the cell's Starbucks. Caffeine equals energy.
Golgi apparatus. That's the Amazon Prime of the cell. It packages, labels, and ships proteins where they need to go.
Endoplasmic reticulum, or ER, is a protein and lipid factory. Rough ER has ribosomes stuck to it, makes proteins. Smooth ER is chill, makes fats and detoxes stuff.
Ribosomes are protein makers. No nonsense. Tiny, tough, and everywhere. Kind of like molecular food trucks, cooking up proteins 24/7.
Lysosomes are the garbage crew. They're filled with enzymes that break down waste, like the janitors of the cell, but with acid.
Phospholipid bilayer is the double layer that makes up your cell membrane. It's got heads that like water and tails that hate it. It's like a molecular sandwich, no mayo.
Concentration gradient just means you've got more stuff on one side of a space than the other. Like when someone sprays cologne in one corner and it slowly spreads.
Dynamic equilibrium means molecules are still moving, but everything's balanced. It's like people dancing on both sides of the room. Nobody's leaving, but the energy's real.
Passive transport? No energy required, like drifting downstream. Active transport? You need ATP. You're swimming upstream and it's leg day. Osmosis equals diffusion of water. That's it. Water moves to where there's more salt, trying to balance things out. Simple diffusion moves small, non-polar stuff straight across the membrane. Easy breezy. Facilitated diffusion? That needs a protein helper. It's like Uber for bigger molecules.
Hypotonic solution? Water moves into the cell, it swells.
Hypertonic? Water moves out, cell shrinks.
Isotonic, balanced, cell just chills.
Plasmolysis happens when a plant cell loses too much water. The membrane peels away from the wall. Cytolysis is when an animal cell takes in too much water and literally explodes. Oof.
Molecular genetics. DNA equals deoxyribonucleic acid. It's your biological blueprint. All your traits are encoded in that double helix spiral.
Nucleotides are the building blocks of DNA. Each one has 3 parts, a sugar, a phosphate, and a nitrogenous base, A, T, C, or G. Watson and Crick built the first accurate DNA model, but Rosalind Franklin? She took the X-ray that showed the double helix. Total legend, underrated.
Those bases pair up, A with TC with G. That's called Chargaff's rule, and it's non-negotiable like biological shipping pairs. DNA replication is the process of copying DNA before cell division. DNA polymerase is the enzyme that adds new nucleotides. Replication fork? That's where the strand splits open like a zipper.
RNA is DNA's single-stranded cousin. It helps make proteins. mRNA carries the code, tRNA brings amino acids, rRNA builds the ribosome.
Transcription is when DNA is copied into mRNA. Translation is when that mRNA is read to build a protein. Together, that's protein synthesis. Okay, so now we're stepping into the part of biology where things get personal. Like, this is why your ears stick out, or why you can roll your tongue but your cousin can't. Welcome to genetics, the study of how traits get passed from parents to kids. Welcome to Unit 6, Inheritance. Let's dive into the fascinating world of genetic inheritance and see how traits are passed down from one generation to the next.
And we owe all this to the original plant dad, Gregor Mendel. The dude looked at pea plants and figured out how traits move through generations before DNA was even a concept. Total legend.
Let's break it down. A trait is something you can see, like eye-colored dimples or the tragic inability to grow a mustache. A gene is the chunk of DNA that controls that trait, and an allele is the version of the gene you get, like getting blue eyes versus brown eyes.
Now, your genotype is the actual combo of alleles you've got. That's like your biological code. And your phenotype, that's how the code shows up. So if your genotype is BBE and brown is dominant, your phenotype is brown eyes.
Homologous chromosomes are like a matching pair of shoes. Same size, same layout, one from mom, one from dad. But even though they match, they might carry different alleles.
If both alleles are the same, like BB or BB, that's homozygous. If they're different, like BB, that's heterozygous. It's like having one blue sock and one red sock. Same function, different style.
Now let's talk cell drama. Sister chromatids are identical copies of a chromosome, two halves of a duplicated file, held together by a centromere like twins in matching jumpsuits.
Then there's DNA polymerase. That's the enzyme that builds new strands of DNA during copying. It's like the DNA version of autocorrect, except it actually works.
Genetic engineering is when we go full sci-fi and directly change an organism's DNA on purpose, like giving a tomato the shelf life of a Twinkie.
And when something goes sideways during DNA copying, that's a chromosomal mutation. We're not talking about a typo, we're talking about rearranging entire paragraphs of genetic code. Could be harmless or chaos.
Okay, now on to the big matchups. A monohybrid cross is when you're tracking one trait. It's like betting on eye color. A dihybrid cross tracks two traits, eye color and height. Now you're playing with combos.
To figure out genetic outcomes, we use a Punnett square. It's a simple grid that predicts the probability of genotypes and phenotypes. Think of it as the fantasy football draft of genetics.
And finally, gametes are your sex cells, sperm and eggs. Each one carries half the genetic code. Put two together and boom, new life, new traits, new drama. Welcome to Unit 7, Cell Cycle and Mitosis. This section will take about 1 minute, give or take a few seconds. All right, class, imagine your body's cells are on a non-stop copy machine schedule. Like they're working the overnight shift at a print shop that never closes. That's the cell cycle. The process of a cell growing, preparing, dividing, and repeating.
And when we say cell division, we're talking about one cell becoming two. Not like cloning a pizza, but close. You start with one cell, end with two identical ones. If that sounds suspiciously like copying homework, yeah, it kind of is.
When cells divide this way, it's called mitosis. That's when one cell creates two daughter cells that are genetically identical. No weird mutations, no funky off-brand copies, just the same cell, now doubled.
But before mitosis even starts, we have interphase, aka the cell's prep zone. In G1 phase, the cell grows, think hitting the gym. In S phase, it copies its DNA like saving a backup file. In G2 phase, it gets ready for the big split. Final checks, stretch break, caffeine, the works.
Then, mitosis happens in 4 fast but dramatic stages. Prophase, DNA gets its act together and coils into chromosomes. The nuclear envelope dissolves like peace out. Metaphase, chromosomes line up in the middle of the cell, like students lining up for roll call. Anaphase, the identical copies, sister chromatids, get pulled apart like a messy group project, splitting up the slides. Telophase, new nuclei start to form. It's the let's go our separate ways phase.
Now about those terms. Chromatin is DNA in lazy mode, uncoiled and chill, floating around during interphase. Chromosomes are DNA in boss mode, tightly coiled and ready for business during mitosis. Sister chromatids, identical twins joined at the center, about to be ripped apart. Brutal.
Once mitosis wraps, we move to cytokinesis, the actual physical cell split. In animal cells, they pinch in with a cleavage furrow, like tightening a hoodie string. In plant cells, they build a cell plate down the middle, construction site vibes.
Each new cell is diploid, which means it's got 2 full sets of chromosomes, just like the original. And since they're somatic cells, they're not for making babies. These are regular body cells, skin, liver, hair, all that jazz.
Okay, if biology was a streaming series, Unit 8 would be the part where the show gets deep. Like, how did we even get here? Why do whales have hip bones? Why do some birds lose their ability to fly and settle for waddling?
This is the evolution unit, people, where science goes full detective mode. We're following the trail of fossils, comparing DNA, and basically solving life's ultimate mystery. How did we go from single-celled blobs to TikTok dancers? Let's start from the beginning. A prokaryote is a super simple, single-celled organism. No nucleus, no organelles, just raw, unfiltered life energy. Like the Nokia flip phone of biology. Indestructible and basic, but it gets the job done. Think bacteria.
A eukaryote is the glow-up. It's got a nucleus, organelles, and it's been upgraded like 20 software versions past prokaryotes. Plants, animals, fungi, we're all eukaryotes, like the difference between Pong and a PS5.
Evolution is the process where species change over time. It's not about turning monkeys into humans overnight, it's about tiny changes stacking up over generations. Kinda like how memes evolve, except with survival on the line.
A homologous structure is like a reused design template. It's a body part that different species share but use in different ways. Like a bat wing, a human arm, and a whale flipper. They've all got the same bones, just built for different functions. Proof that we had a common ancestor.
Vestigial structures are the biological equivalent of a landline phone in your kitchen. Used to be important now. Just there. Like the human appendix or wisdom teeth. They're leftovers from evolution's past lives.
A half-life is the amount of time it takes for half of a radioactive substance to break down. It's how we do radioactive dating to figure out how old fossils are. Basically, nature's built-in countdown clock, but for atoms.
Radioactive dating is how we determine the age of fossils using half-lives. It's science's version of carbon dating your ex's playlist to figure out when they peaked, only way more accurate.
Variation is just differences between individuals in a population, like some people can digest dairy and some people, well, shouldn't. These differences are the raw material evolution works with.
Natural selection is the main engine of evolution. It's the idea that organisms with traits better suited to their environment survive, have babies, and pass on those traits. It's like biology's version of if you know, you know.
Artificial selection is when humans play favorites. We breed dogs to be fluffier, or corn to be sweeter. It's evolution directed by us, basically nature with a casting director.
Acquired characteristics are traits you pick up during life, like muscles from lifting or a scar from falling off your bike. They're not in your DNA and you can't pass them on. They're like unlocked achievements, just for you.
Inheritance is the stuff you do pass down through your genes. Hair color, dimples, weird ear shapes. It's like a hand-me-down sweater, but made of DNA.
Survival of the fittest doesn't mean gym strength. It means the best fit for the environment. Like, in a desert, the fittest might be a tiny lizard who barely drinks water. The key word is fit, not jacked.
A mutation is a change in DNA. It might be random, or caused by radiation, or a copying error. Some are bad, some are good, most are just neutral. But it's how new traits enter the gene pool.
Sexual selection is when traits evolve because they're attractive to mates, like peacock's ridiculous tail feathers or deep voices. Basically evolution, but make it hot.
Sexual reproduction involves two parents mixing genes. The result? New combinations, more variation. Nature's genetic mashup session.
Asexual reproduction is cloning. One parent, identical copy. Fast, efficient, zero drama, but way less variety. It's like copy-pasting a document instead of writing something new.
Microevolution equals small changes in a population, like bacteria becoming resistant to antibiotics. It's local, short-term evolution, kind of like new slang showing up every few years.
Macroevolution is the big stuff, like fish evolving into land animals. It's the reason birds and dinosaurs are related. Yep, birds are dinosaurs. Jurassic Park lied to no one.
A species is a group of organisms that can mate and have babies that can also have babies. If they can't do that, not the same species. Sorry, Shrek.
Speciation is when one species splits into two, usually because of isolation, like one group gets stuck on an island, adapts to that island, and bam! New species, who dis?
Disruptive selection favors both extremes over the middle. It's like loving spicy food and super bland food, but ignoring everything in between. You either go ghost pepper or plain rice.
Directional selection is when one extreme is favored. Like if longer legs mean better survival, the species just starts growing NBA players over time.
Stabilizing selection favors the average, like babies being born not too big, not too small, just right. It's Goldilocks evolution.
The gene pool is all the genetic material in a population. It's like a big pot of DNA soup. Everyone's tossing their alleles in there.
Gene flow happens when individuals move between populations and mix their genes. It's genetic exchange, like new kids moving into your school with different memes and changing the culture.
Genetic drift is when allele frequencies change by random chance. Not survival, not selection, just luck. It's evolution's version of rolling dice.
Geographic isolation happens when populations are separated by physical barriers, mountains, rivers, oceans, no texting, no travel. Eventually, they evolve differently.
Behavioral isolation means two groups don't recognize each other's mating behavior. Like one dances, one sings. Neither is into it. No spark equals no babies.
Mechanical isolation is when the body parts just don't work together. Literally, like trying to plug in a USB the wrong way. It's not happening.
Temporal isolation equals wrong timing. If one species mates in spring and the other in winter, they're never on the same schedule. Netflix says, "You're not compatible."
The fossil record is like Earth's scrapbook. It shows us what used to be here, how species changed, and who's extinct. Bones don't lie.
A cladogram is a diagram showing shared traits between species. It's like a flowchart for evolution, but make it stylish.
A phylogenetic tree is a visual of evolutionary relationships over time. Think of it like a family tree, but your cousins are birds and sea stars.
Phylogeny is the study of those evolutionary connections. Who descended from who, what traits got passed down, and where things split off. It's like following the DNA drama across millions of years.
Oh! That was Unit 8. Fully defined, fully explained, and fully ready for your test and the final boss battle of your biology year.
And remember, biology isn't just about facts, it's about figuring out how life pulled off the biggest flex in the universe.
Study hard, evolve faster, pass everything. Okay, so, you know how in sci-fi movies, scientists are always playing God with genetics? Yeah, that's real now, and it's called biotechnology.
Yep. Welcome to the unit where biology gets turbocharged by tech. We're talking gene editing, glow-in-the-dark animals, and designer corn. This isn't science fiction. It's Tuesday in a genetics lab.
Let's start with the big one, genetic engineering. That's when scientists manipulate DNA on purpose. Want to make tomatoes that don't rot? Or bacteria that eat oil spills? That's genetic engineering. It's like editing your biology Google Doc, just with way more consequences.
CRISPR is the tool that makes it happen. It's basically molecular scissors. You can cut out parts of DNA and swap them like Pokemon cards. It's precise, powerful, and just a little terrifying. Kind of like when your younger cousin gets admin privileges.
A transgenic organism is one that has DNA from another species, like goats that make spider silk in their milk, or pigs that glow. It's like Frankenstein, but with better lighting and a grant from the government.
A plasmid is a tiny ring of DNA found in bacteria. We use them like USB drives to transfer genes. Load up the plasmid with a new gene, stick it in the bacteria, and boom, it starts doing your bidding, like a microscopic intern.
A vector is anything that delivers genes into a cell. Could be a plasmid, a virus, or some other tricked-out delivery system. Think Uber Eats for genes, but instead of food, you're dropping off blueprints for glowing frogs.
Recombinant DNA means DNA from two different sources combined into one, like building a playlist with songs from two totally different genres. Only now your playlist might cure cancer or make strawberries frost-resistant.
Your phenotype is what you actually look like, based on your genes. Like you might have the genes for blue eyes, but your phenotype is blue eyes. It's the final product. The cake, not the recipe.
Selective breeding is the OG version of genetic engineering. Instead of editing DNA, humans pick which parents will make the cutest, smartest, fluffiest babies. That's how we got pugs and corgi butts. You're welcome, world.
Gene splicing is exactly what it sounds like, cutting and pasting sections of DNA. It's scissors and glue for genetic code. Only instead of an art project, you're making insulin or turning rice into a vitamin A supplement. Casual.
And of course, we've got mutation, a change in DNA. It can happen randomly, or we can do it on purpose in a lab. Mutations are the source of all genetic diversity. Sometimes they create superheroes. Other times, lactose intolerance. Roll the dice.
So yeah, biotechnology is like the ultimate science toolkit. It's powerful, it's weird, and it's changing life as we know it, literally.
And now you officially understand the language of genetic hackers, DNA remixers, and science rebels everywhere.
Stay curious.
Stay scientific.